Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Oncology and Immuno-Oncology Precision Medicines, from Hit Finding to Preclinical Candidate Selection. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Oncology and HitGen who developed the content for this presentation. Oncology is a biopharmaceutical company which aims at innovation. Its mission is to discover innovative and effective new therapies for the benefit of patients suffering from cancer and serious illnesses with unmet medical needs. Oncology service is an integrated solutions provider in drug discovery. Next, I'd like to introduce you to HitGen, which is a small molecule drug discovery and development company which has established an industry-leading platform for early-stage drug discovery research centered on DNA-encoded chemical libraries. HitGen is a premier provider of DELs and DEL technology, collaborating with multiple pharmaceutical and biotech companies, foundations, and research institutes to discover and develop approaches for novel medicines. Now it's my pleasure to introduce you to one of your speakers for today's webinar, and she is Emily Fang, who is the Director of Business Development for Integrated Drug Discovery at Oncology's Services Business Unit, and also the Strategic Partnerships in the DRIVE Consortium. She brings a decade's experience in international business development, specializing in the early discovery space, and having worked in diverse companies ranging from startups to multinational organizations in many countries. Now, it's my pleasure to pass over the mic to Emily. So, Emily, when you're ready, you may begin.
Thank you. Thank you very much, Sonia. I'm very happy, and on behalf of Oncology Services and the DRIVE Consortium, I'm very pleased to be moderating this special webinar jointly presented by two of our collaboration partners, Oncology and HitGen. So next, please. Next, please, Sonia. Thank you. I'd like to first take a couple of minutes to share with you really the inspiration and the unique concept behind this consortium that we have constructed in the early discovery space. And at this time, it's focusing on small molecule discovery and development. So both Oncology's discovery team, and then you will hear in a few minutes from the scientific leadership at HitGen, they're bringing together decades of experience in the big pharma, in the pharmaceutical industry. And it is actually with this background and this collective experience that they're also acutely aware of the challenges that we all continue to face in this industry. And this is how Oncology is presenting a model and, of course, a solution on how to more effectively leverage external sources of innovation, of the latest technologies, and most importantly, the experts and the expertise. That's really where the value lies. And particularly in early discovery, technologies are constantly changing, improving, advancing. So these traditional conventional approaches of acquisitions and integration and working with CRO service companies in these silo modes and isolated fashion is really no longer practical. And it's, as far as timeline and resources, is no longer tenable. So what we're doing is we're bringing together really an incredibly unique network of technology specialist companies who are all individually experts and leaders in their respective domains. And where we're creating value is what I like to call really having the best of both worlds in externalizing discovery to operate this in a structure to allow, for example, a biotech company, biopharma, or pharmaceutical company to access these innovations and technologies in a more flexible way, fast faster, of course, with a lower, essentially lower barrier of entry, and in a fashion that suits each individual company's specific objectives and strategies. So Oncodesign provides this on the other side of the coin, one single centralized management umbrella that includes project management, operational, and administrative management. So next, please. So these partners within the DRIVE consortium, they each have complementary expertise and capabilities. We're constantly closely collaborating and communicating, so there's continuity in the project, and really importantly in understanding what each project needs, because each project is special. And I think as anyone maybe here around the table that has that experience in the trenches managing multiple technology collaborations and relationships will attest to, it's really the reactivity and proactivity between the communication that's really critical in delivering the ultimate drug candidates. So what we're really hoping to bring to the table and to the discussion today is this collaborative mindset that you can trust in the scientific excellence and the quality, and we're bringing to you this commitment to success in everyone's research program to bring innovative therapeutics onto the market. So next, please. And the solutions that we're bringing through the DRIVE integrated discovery platform covers the entire drug discovery and development continuum. And this is really powered by Oncodesign's deep experience in the pharmacology, in the biology, in major therapeutic areas, including oncology, immuno-oncology, inflammation, and infectious diseases. So with that, today, you will hear more in depth about a few of the specific technologies and translational toolbox within this continuum from Oncodesign and from our partner, HitGen. So with that, I'd like to introduce the first speaker from HitGen. So Barry is, of course, Dr. Barry Morgan is the Chief Scientific Officer for HitGen, where he focuses on developing and applying DNA-encoded chemistry technology to early-stage small molecule drug discovery. So Barry has over 40 years of experience in research and development in pharmaceutical and biotechnology industries in the UK, in France, in the US. He was Vice President, Molecular Discovery, and Site and Business Head at GlaxoSmithKline at Boston between 2007 and 2012. He was previously the Senior Vice President for Chemistry and Discovery Sciences at Precis Pharmaceuticals, where he was a primary inventor of DNA-encoded library DEL technology. So Barry has presented at invited seminars at over 100 academic and industry symposia in Europe, in the US, and China. He has authored over 100 publications and posters as an inventor on more than 40 patents in the areas of drug discovery. He has contributed to over 20 drug development candidates in a range of therapeutic areas, of which more than 10 have advanced to clinical development. And Barry has a faculty position at the Institute of Molecular Medicine at the University of Texas at Health Sciences at Houston, Texas, where he focuses on collaboration, applying DEL technology to novel drug targets in academia and the economics of drug discovery and development. So it's with my great pleasure, I will introduce the first presentation from Barry at HitGen.
Thank you, Emily. Let me just share my screen with you folks. I hope you can see my screen and I hope you can all hear me. So again, thank you for taking the time to attend our webinar. What I'm going to do in the next 20 minutes or so is give you a little bit of a background on how DNA-encoded chemistry evolved, show an example of a collaboration that we carried out a few years ago. It's now published with our Pfizer colleagues, and summarize HitGen briefly. So let's get started. I think that early stage discovery is an exercise in managing serendipity. So I'm going to start with this proposition that hit finding is an exercise in improving the economics and perhaps more importantly the timeline in navigating chemical space to find a hit matter. And when we started looking at this concept back in 2001, we wanted to disrupt the model which was current at that time. That is a conventional high throughput screen followed by cycles of medicinal chemistry. That was a fairly effective model, but it was kind of expensive and kind of fast. And using the concept of disruptive technologies, initially described by Clay Christensen here, we contemplated that if we could find a technology that allows inexpensive synthesis and interrogation of very large numbers of compounds, we could bring disruptive innovation to early stage small molecule drug discovery. So we started considering this. We didn't have to look too far because this paper, published in 1992 by these two gentlemen, provided the background for DNA-encoded chemistry. And I want to just look at this sentence from the paper, and because it really says it all about the technology even now. So the nature of the chemical structure is decoded by sequencing a nucleotide tag. So as I'm going to show you, the only thing we measure in this technology is actually the frequency of occurrence of certain oligonucleotide sequences coming through a screen by selection, and then we translate that back into a related set of encoded chemistries and some potential products from those encoded chemistries, and I'm going to attempt to show you that. So a brief history, as I already mentioned, the seminal publication of Brenner and Lerner defined the technology. A few years later, Kinoshita and Nishiaki at a small university north of Tokyo published a two-page paper showing that enzymatic ligation was key to assembling the encoding DNA. And then not much happened for some years until there was a renewed interest due to some publications by a group led by David Liu at Harvard on DNA templated chemistry. At the time, I was at a small company called Tracis, and we felt we had a much more pragmatic approach to the assembly of these libraries. We published that in a 2009 paper, which I'm going to show you in a moment. And currently, in 2021, it's pretty much broadly accepted and utilized within both the pharma and biotech sector and in academia, either internally or with providers such as HitGen. Those of you who are interested in other ways of DNA encoding, there's a Wikipedia site which explains those. I'm just going to focus on using the DNA as a barcode. And this is the paper we published. I'm not going to be able to go into too many details today due to time constraints. All of the details are still in this paper and in this reference here by Rob Goodnow and his colleagues, some of the hit matter that's been described in the literature. So let's take a look at the fundamentals of the technology. First of all, you design and assemble billion component DNA encoded libraries. I'm going to show you one example of that. Then you actually select against the library using a selection methodology, rather similar to phage display, against a target, usually a tagged protein target. Hopefully some species in the library will bind to the target. So you collect that fraction, amplify and sequence the DNA, and come up with these cubic plots. We use a program called Data Warrior. I'm going to show you a couple of examples of this and try and explain at least some of the basic concepts from these lines in this of dots in these cubic plots. We call them features. You can identify what chemical species to make. It's fairly intuitive, I believe, not that complicated. And from that, we decide on some compounds to synthesize without the DNA tag and characterize them from a biophysical or biochemical perspective. So these are the steps that are involved. They are described in the paper I just referenced, but let's take a little look in some more detail. So I'm going to use this library to explain some aspects of the technology. We made this library back in Tracis in 2004, I believe. It was the 16th library we made then. And as you can see, it's a 4-cycle library around a non-coded triazine scaffold. So I'm going to use this library to explain just a few concepts. The libraries are made by split and pool, and then you're doing sequences of encoding. You can see little DNA codons here, and then chemistry that's specifically associated with those codons. Okay, let's take a little bit about some of the concepts of the technology. You mentioned, I mentioned that we use features, that is selected sets of encoded syntheses that share one or more component of the combinatorial chemistry to identify the hits in this technology. And what we're looking at here is a real example of a selection against DEL16. This is a 3-cycle library, so each cycle of chemistry can be aligned along one of the axes of the cube. So each encoded synthesis in this library has a unique location in this cubic space. a little bit of statistics. So when you interrogate the outcome from a selection, there are two numbers you need to be concerned about. One is what is the total number of potential species that you're interrogating, and this library had 800 million species, let's call it 1 billion. And then the second number is the number of sequences you're using to interrogate the library. When we did this selection, we were using 100,000 species, 100,000 sequences per selection. So if you drop those two numbers into a Poisson probability function, you get the outcome that the chance of randomly seeing a specific sequence twice is 500,000 times less likely than seeing it once. We don't have time today to go into the depths of Poisson probability analysis. So let's just take this concept as being true. So that means that a selection that allows enrichment under these conditions to a situation where we get 2 or more copies, we should see that as highly significant and easily identifiable above background random DNA noise, which in this case are all the single species. So what I'm going to do is, looking at this cube, is remove all the times when the sequences only occurred once. And this is a real example, as I mentioned, and what you see is we see this line of larger spheres. Now, the diameter of these spheres is related to the number of sequence reads for that specific encoded synthesis. And when we saw this, for some reason, can't quite remember why, we called it a feature. And because it appeared in a line, we called it a line feature. And pretty much most people still use those terms and use these cubic plots to explain the technology. So what I'm going to do now is look at a more specific example, which was recently published by us and our colleagues, Pfizer in this case. And I'd like to acknowledge Pfizer for this work, and in particular, Dr. Pei Kung, who was the project leader at Pfizer. And as you can see, this is published last year. So let's take a look at this. So the target here was an acetyltransferase, NAA50, and this enzyme transfers an acetyl group from acetyl coenzyme A to the N-terminus of a set of peptide substrates, and then leaves the coenzyme A without the acetyl group. And there was a crystal structure of this. One of the great aspects of success of this technology is our understanding of the biophysics of the target, and anything we can find on that. And there was a literature report saying that the cofactor, acetyl coenzyme A in this case, was required to induce a substrate binding pocket. The substrates, of course, being the peptides. So let's take a look. Also in the literature was this bi-substrate inhibitor, which was essentially a conjugate of acetyl coenzyme A plus the peptide substrate with this thioether linkage. This was quite effective inhibitor of NAA50, but not a particularly good starting point for a drug discovery attempt. You can see that the bi-substrate inhibitor was quite a potent inhibitor, 16 nanomolar, but neither the product, that is coenzyme A, of the cofactor, the peptide substrate itself was a particularly good ligand. So what we set out to do was to find ligands that bound to NAA50 using this information. So what we always do when we start a collaboration is to set up identify a set of selections with our collaborators. And I'm going to go through those now. So we always, first of all, use the protein. It's a tagged version of the protein. We need the tag to be able to pull it down. And then knowing the mechanism of the target, so we'll occupy the cofactor with that site with acetyl coenzyme A. Then we occupy the cofactor site with the product when the acetyl group has been transferred. In this case, coenzyme A. We wanted to see which species were competitive with a known bi-substrate inhibitor. So we carried out a selection in the presence of the bi-substrate inhibitor. And lastly, we want to make sure we don't look at artifacts. So we always do a no target control. In other words, a selection where we don't add the target. In this case, we never saw any features in the no target control. So I'm only going to look at these first 4 selection conditions. Okay. And we're going to look at one of our HitGen libraries which showed up in this selection, DEL951. We have something like 1,600 libraries currently. And this library, 5 synthetic steps, 3 encoded cycles, essentially it was adding 2 sets of amine reacting groups to this sequentially deprotected diamino acid. So this was a scaffold-based library. About 2/3 of our libraries are scaffold-based, and the other third are libraries where we actually construct the scaffold as a part of the library. And in this case, we added in the cycles 2 and 3 the same set of acids, aldehydes, sulfonyl chlorides, and isocyanates to this sequentially deprotected diamino acid. You can see in one case, we removed a protecting group. I believe it was an FMOC group. And in the other case, we introduced the amine group by reducing this azide function. I don't really have time to go into the concept of nulls, but nulls in our encoded libraries are there to show the relevance of a particular set of selections. So you can find the description of nulls in the paper that I just mentioned. Okay, so let's take a look at this library and the selection conditions that I described. So here's the selection against the APO enzyme. You can see, like just as I showed you in the previous example, there are sets of lines running in several directions. So that was quite interesting. So we were able to identify features. It's always a good start when you can see features. However, when we added acetyl coenzyme A to the selection, you can see that we got much more higher pronounced features. You can see also that some of them were changed, so that these lines in the APO enzyme disappeared in the case of adding acetyl coenzyme A, whereas this set of lines were considerably amplified. You'll notice there is some features which are maintained. Look in particular at these 2 large spheres here. Remember that the diameter of the sphere relates to the number of screen reads, accounting for that in the sequencing output. You can see that these 2 are actually there in the APO enzyme. So it's interesting that they were amplified when we added acetyl coenzyme A, but perhaps if you recall that the literature told us that this cofactor was necessary to induce a substrate binding site, that might be explained by this amplification. Okay, in the third case, we did the selection where we added the cofactor without the acetyl group. And you can see that we got a pretty much reproduction of the situation with acetyl coenzyme A, but not quite as amplified. And that might also have been suggested by the relatively low affinity of coenzyme A itself. So these 3 were consistent now with an identification of several features which were perhaps acting at the substrate binding site, which was defined by adding the cofactor. Okay, so what were these large species? The largest sphere here had over 2,000 copy reads, which is a pretty high number, and was in fact this encoded synthesis. This scaffold here, this thiazole racemized at this center during the synthesis of the library. So this was a pair of diastereoisomers. Let's take a look at the last selection condition where we added the bisubstrate inhibitor. And as you can see, in this case, it competed away all of these features, suggesting strongly that wherever this bisubstrate inhibitor was interacting, then the site of these features were obliterated, suggesting that they were probably interacting at the substrate site, as we already suspected from the use of the acetyl coenzyme A selection. So this is how we design a set of parallel selection experiments and use those parallel selection experiments to interpret the mechanism of the potential features. Okay, let's take a look at how we characterize these compounds. So here, and we're using thermal shift data. So here you can see the thermal shift of the apoprotein in the presence of the bisubstrate inhibitor. Quite a dramatic thermal shift of 19+ degrees, which is a pretty high shift for those of you who are familiar with this technology. Let's take a look at the compound that we identified. So this is the pair of diastereoisomers, and you can see that they shifted about 9 degrees, and if you then added acetyl coenzyme A, there was a further shift to a total of over 21 degrees, which was on the same scale as the bisubstrate inhibitor again suggesting that the mechanism of this species mimicked, plus acetyl coenzyme A, of course, how the bisubstrate inhibitor interacted with the target. And all the data were now coming together that this species almost certainly interacted in the substrate site induced by acetyl coenzyme A. So indeed, this mixture of diastereomers was a fairly potent inhibitor of NAA50 with a 13 nanomolar IC50. Our colleagues at Pfizer synthesized both of the diastereoisomers, and the activity was found to reside in the cis isomer, the trans isomer being considerably less active and maybe inactive, depending how pure it was in terms of cis versus trans. So this was the lead that we found from this selection campaign. And let's take a look now at just the crystal structure of this cis isomer, in the target. And you can see that indeed it binds in the substrate binding site. And this was a co-crystal structure in the presence of coenzyme A. So as the selection pattern suggested, we found an inhibitor which bound in the substrate binding site, and that substrate binding site was defined by the presence of the cofactor. So when you see these type of mechanistic approvals, if you will, of a selection campaign, you can be fairly confident that you're going to get a good outcome from the selection. Just one last point. So you remember there were two fairly large spheres in parallel line features in this particular selection. This was the highest one. There was one almost as large right next to it. What was that one? Well, that happened to be the oxazole containing scaffold. And again, the oxazole was almost equiactive to the thiazole. Again, another thing to look for in the outcome from an encoded library selection is an internal structure activity, either within a library, like in this case, or between several libraries, and we frequently see that. So that's the description I'd like to explain. There is considerably more detail mentioned in the publication I showed earlier in this presentation. So that's the outcome. Let me describe now a little bit about HitGen. So HitGen was founded in 2012, and our laboratories are built a few years ago now, are located just outside of Chengdu, China, Tianfu International Biotown. We also have subsidiaries in Houston, Texas, serving the US, and more recently in Cambridge, UK. The company currently has over 500 employees, mostly in research and development, and over 200 chemists. As Emily mentioned, we have a platform focused on the design, assembly, and interrogation of high-fidelity de-encoded libraries. Currently, we have over 1 trillion encoded syntheses in our small molecule DEL pools. We also have an electrophile DEL pool, which is growing. We have over 20 billion encoded species in the electrophile DELs, and our mission is to be a leading provider in progressing early-stage projects from target to IND-capable asset. And what I show you here is our robot setup on the top floor of our building, where we currently make all our libraries. We have a wide range of partners. I show an alphabetical list of them here, of the folks who we have announced as partners, covering North America, Europe, Asia, Australia, and Africa. And as you can see, we have large pharma companies, small pharma companies, biotechs, foundations, and an increasing number of academic partners. We have business models which work with all of these groups, and we'd be more than happy to talk with you about a model to suit your purpose. So screening by selection, this is the result of our experience over the past 3 or 4 years. In particular, the last 2 years, we've been quite successful with licensing some assets at least once a month. These are the classes we've looked at. Typically, when people get interested in encoded libraries, they typically give us the harder drug targets. So as you can see, the most popular class we've been given as collaborations are protein-protein interactions. we've had some reasonable success with that class of target, but I'm not going to pretend that this is a panacea for hard-to-drug targets. The better you understand the biophysics of your protein and the biochemistry, how you're going to look at the outcome, the more likely we are to succeed working with you. The second class, perhaps, as you can see, are kinases, but we've looked at quite a range of class of targets. So I hope I've been able to explain some of the fairly basic fundamentals of the encoded libraries. I'd be more than happy to discuss any details with you. You'll see my email in a minute. And again, I'd like to thank you for listening. We really are beginning to solve a lot of the problems of encoded libraries. And I'd like to use this quote by Sidney Brenner, that progress in science depends on applying new techniques in new areas. Virtually all of the DNA handling methodologies I described today were not developed at HitGen. We developed the way of making the libraries, and we think that this is really a way of managing serendipity in chemical space. And with that, I'll pass the slides back for the next presenter.
So now I'm really pleased to introduce the next speaker, who is Olivier Duchon. He's the head of in vivo sciences at Oncodesign Service Business Units. And he was, of course, at the founding of the company, and for the last 23 years has contributed significantly to the development and innovation in preclinical models and pharmacoimaging. Olivier is also the coordinator of Immuno, which is a French consortium dedicated to the development, the characterization, and the commercialization of new preclinical models in oncology, including PDX models and humanized mouse models. So it gives me great pleasure to give the platform back to Olivier for his presentation.
Yeah, thank you, Emily, for this introduction. So I think that you are now seeing my screen. Thank you all for attending this meeting, in which I can try to show you and perhaps to convince you that we can use patient-derived xenograft models, PDX, in a novel, holistic and integrative approach for drug discovery and drug development. So the main reasons of why developing new models in cancer is based on the fact that the success rate in the development of compounds in oncology is very bad. So because there's only 6 to 8% of the compounds entering into the clinic that are reaching the market. So it's a very poor efficiency in drug selection in our preclinical and clinical process selection. So that's why at Oncodesign, we are always thinking that we can increase the efficiency of the preclinical models that we are using in drug selection in the preclinical proof of concept. So, but the major difficulty in oncology for sure is to modelize the diversity of the cancer pathologies in order to be able to better predict the human clinical response. So, and in fact, at Oncodesign, we have made the choice since many years to develop innovative models in a collaborative manner. And especially the PDX that we developed in collaboration with pharmaceutical companies, with CROs, and with many different academic labs that are working all together in order to increase the availability and for research and the efficiency of our models. So, and for the ones that are not completely aware about a PDX, a PDX is an in vivo model that is developed by engrafting patient tumor biopsy directly in mice without establishing tumor cell lines in vitro. And based on that, okay, so that a PDX become a model only when annotated, and the annotations are based on different characterizations. So for sure, the histology, the gene sequencing, the gene expression, the SNP polymorphism, etc., etc. that we need to put around the PDX to completely characterize the models. And with Oncodesign and with our partners of the Immuno consortium, we are collaborating with many different groups in the world to define some international standards of this annotation that we have to put around the PDX to better use these models. So the next one, please. So, but developing models, so it's interesting. So it's a good, a nice adventure, but for doing what? So now I will try to show you some applications of this type of models in the different steps of preclinical drug development. So the first one is to show you some application of the target validation and candidate selection. So next, please. And as mentioned before, the main advantage of the PDX is to capture the heterogeneity of the cancer diversity, for sure, between cancer indications, but also in a cancer, in specific cancer indication. Here you can see in this example of our PDX models in breast cancer, that we have succeeded in the establishment of PDX models in each of the 4 main subgroups of the breast cancer pathology. And as I mentioned before, so we have characterized these models regarding the histology, the genomic profile, just to be sure that the tumors that grow in mice are completely similar to the ones that are in the patients. So next, please. And the same with the RNA-seq analysis that we perform in the patient tumor and in the early and late passages in mice. And you can see in the figure here that all the samples coming from the same patients clustered together. So next, please. But for sure, using in vivo models, you may imagine, is not so efficient and not so easy to use for the early stage of preclinical development, especially when we are on the field of screening a large panel of molecules to select the better leads. So using vivo models is not so easy. And that's why we have decided to develop cell lines derived from the PDX established in mice. So in this case, so we have succeeded in the establishment of 8 cell lines from the 35 PDX models that we have developed for breast cancer in mice. And you can see here that we also obtain cell lines that represent all of the 4 major subgroups of breast cancer. And of course, these cell lines are also characterized with histological markers and SNP array to be sure that they correspond to the initial patient tumors. And these cell lines could be cultured with or without corresponding fibroblasts that could be extracted from the original tumors. And then next slide, please. For the tumors with which we did not succeed in the establishment of cell lines, so we have developed with our Immuno partners some ex vivo assays that are running with 2D or 3D systems in order to test many compounds in a large panel of tumors. Here you have an example of 3 compounds that we have tested in 2 different breast PDX models. And you can see that with the first model, we obtained the same results on moderate efficacy of the 3 drugs. And with the second one, the drug one in blue is the most efficient drugs in the second model. So for sure, so we have investigated this drug one in the corresponding in vivo PDX models, and you can see in the left that in blue the drug one is the most efficient drugs in these 2 PDX models, and these PDX are luminal B with cancer. So this helps to demonstrate the efficiency of this compound, of this lead compound in this type of breast tumors. So next, please. But in parallel, we also want to know if this drug one should be also efficient in triple-negative breast cancer, which is a clinical needs. And then for doing that, we use a tissue microarray in which we select the TNBC model that highly express the target, and another one in another indications which did not express the target of the compound. And you can see that we obtained a huge efficiency and demonstrate that tumors with at least 50% of the target cells could respond to the drug one. So this is a very interesting data. But next, please. But in fact, based on these results, so how to know and if all the breast tumors could answer to these treatments. So is there some specific tumors that could better answer than others? Or what about the tumors that are with moderate expression of the target? So for answering this question, next please. So we are now routinely using the concept of single mouse preclinical trial in which we are putting only one mouse per model and per experimental conditions. So meaning that we are able to test several compounds in a large panel of tumor models that are all representing a patient. So it's possible to run studies with 30 or 40 different PDX tumors that are representing each different patients. So next, please. And then just for you to show you, so we have tested this drug one in a small panel of 12 different breast PDX models that in which we put 7 to 10 mice per group. And so we have classified the individual response of each mice in 4 categories based on the RECIST criteria, which are the criteria that are used in the clinic to classify the response. And we found that more than 70% of the individual response matched to the median response of each group. But if you pull the 3 category, the complete responder, partial responder, and stable disease in one single responder category, then we obtain 100% of the individual response that could match with the median of the group. This completely demonstrating that this experimental approach could be used to define the tumor that could better respond to one or several compounds. So next, please. And for sure, so we have tried this drug one in a larger panel of breast PDX models. Unfortunately, these data are still confidential and not available for this meeting today, but I have put here another example just to demonstrate the feasibility that we perform with a large panel of approximately 30 colon PDX models in which we apply FOLFOX combination in all the models, and you can see that this combo increase the number of tumors that could answer as compared to the monotherapy. And this clearly corresponds to what we are seeing with this combination in the clinic in terms of survival of the patients. And as you can imagine, so the combination of these pharmacological informations on all these models with the annotation that we have for all the PDX associated with the artificial intelligence platform that we have developed, clearly benefits in the identification of biomarkers of drug efficacy and also tumor and patient stratifications. And also all these studies so could help us to better position the drug into the clinic. And next, please. And based on all these results, so this drug one was approved in the clinic and for the treatment of patients with advanced breast cancer after 2 prior chemotherapies. But based on the preclinical studies and specifically the in vitro mechanism of action studies that we performed during the preclinical development of this compound, so we hypothesized that this compound should affect the cellular differentiation pathway via some cell cycle checkpoints. And this led us to ask the question on how to succeed if we combine this drug one with a anti-cancer agent that could have an effect on this on the cell cycle. So, and that was the rationale of the initiation of the new study, aiming at investigating triple combination of drug one with palbociclib, a CDK inhibitor, and fulvestrant, an estrogen receptor antagonist. And so we clearly demonstrate in vivo model here, luminal B breast cancer, that we obtain a great synergy of drug one when associated with palbociclib, and that we increase this synergistic effect with the triple combination of drug one with palbociclib and fulvestrant. So this was a great demonstration, but as you probably know, many women that are affected by this type of breast cancer pathology are treated with hormonal therapy and develop hormonal treatment resistance after several months or several years of hormonal treatments. So next, please. That's why we have decided to try to mimic this clinical drug resistance with our model, and we have tried to establish new models that can recapitulate this clinical situation with women escaping the hormonal therapies, and in order to try and to test this drug one in these clinical situations. So, and we have selected one PDX model, a luminal B with PDX model based on the tumor growth characteristics and its response to endocrine therapy. And next, please. And we have defined experimental conditions in which these tumors can grow without estrogen and under treatment with fulvestrant. This is specifically the case of the group 3 in these slides, in which you can see that we obtain a slight delay of the tumor growth, but all the tumors escape these treatments and continue to grow even estrogen deprivation or fulvestrant treatment. And of course, we have characterized this model to be sure that we have a clear fulvestrant resistant model, and you can see here that all the hormone receptors expression are clearly affected by the endocrine therapy, especially in group 3 and 4 that are treated with fulvestrant. Next, please. And also by characterizing these resistant tumors by RNA-seq, and you can see here that all the tumors that are treated with fulvestrant clusterize together, here the cluster 1, and that in this tumor, we clearly see that many genes are upregulated and that these genes are related to endocrine therapy resistance and with KRAS pathways. And next, please. And then after, for sure, we have characterized better this model and confirmed that the tumors can clearly grow without estrogen and with fulvestrant treatment, and there's no effect of the fulvestrant. Even we increase the dose of the compound, we didn't see any effect of the fulvestrant arrest and no effect of the addition of estradiol. This means that we clearly have developed a fulvestrant-resistant breast PDX models that is completely independent of the estrogen. Next, please. And for sure, as you may imagine, so now we are using this resistant model in order to mimic the clinical situation and in order to know how to apply and to use the drug one in this resistant model. Is the drug efficient on this type of tumors? And the other question is how this drug one could restore the fulvestrant initial effect. So this study is still ongoing, so unfortunately I can't show you this final data, perhaps in the next webinar session, if possible. But next slide, please. But for sure, so as always, all models have their limitations, and here, when we develop PDX models, so we engraft human tumors in immunodeficient mice. So next, please. And in this case, how to discriminate and to test and evaluate compounds that are targeting the tumor microenvironment and not the cancer cell itself. So, and next, please. And this is especially the case in the field of immuno-oncology that are targeting on the module the immune cells. So, in fact, it's very easy. Just humanize mice with human immune cells and then engraft PDX tumors in these humanized mice. So it's easy to do. So, and here just to show you that it's completely feasible and it's that is completely to mimic also the patient, the human situation. So we have investigated a large panel of tumors and investigated the immune infiltration in these tumors coming from patients in breast and ovarian cancers, and we selected two tumors in each cancer indications. One that is highly infiltrated with immune cells in the patients, and one that is very poorly infiltrated by immune cells. And then we have engrafted these tumors in humanized mice, and you can see in the right panel in this slide that the tumors that are highly infiltrated in patients are also highly infiltrated in humanized mice. And on the contrary, the tumors that are very poorly infiltrated in patients remain very poorly infiltrated in humanized mice. This clearly demonstrates that this is the tumor cells that are driven the immune infiltration in the tumor. And of course, so we have tried to evaluate the efficacy of pembrolizumab, an anti-PD-1 compound in the most infiltrated PDX tumor in humanized mice, and you can see that we didn't see any efficacy of these compounds in this model. Even the tumor is well infiltrated by immune cells. But I would say it's exactly the similar to the clinic as we as the anti-PD-1 have never demonstrated their clinical efficacy in the triple-negative breast cancer. So next, please. And in order to still continue to increase the efficiency of our model, we are now developing autologous models in which we can have the tumors and the immune cells coming from the same patients. And for doing that, we are using iPS cell technologies and iPS that we can generate with PBMC that are taken from the patients the donor of the tumor, we are able to derive hematopoietic stem cells from the iPS and then inject these hematopoietic stem cells in mice to reconstitute an immune system in mice that are bearing the tumors coming from the same patients. So this is where we are now, and these models are still under development, and we expect to have this model ready to use probably by the end of the year. So this is my last slide, and I hope that these slides will have convince you and that PDX are good models for drug discovery and drug development. And we are still continuing to develop new models and innovate to increase the efficiency of our drug selection in preclinical phase in order to benefit to the patients. Thank you very much, and so open to question now. Thank you.
Well, thank you very much, Barry, Olivier, and Emily, for that very detailed presentation. I hope everyone enjoyed it. We're running a little bit late here, but we're going to try to squeeze in 2 questions. So I'm going to try to encourage the audience to continue sending in their questions by using that questions window. And I know we won't be able to address them now, but we'll be able to follow up with you later on. But right now, Emily, I believe we have a couple of questions that have already come in, so I'll leave it to you, and then I'll come back in about 3 minutes' time. Thank you.
Yes. So I have a question here, first for Barry. So the question is, what about the proteins that may include a DNA binding domain? Does that pose a problem when applying the DEL technology?
Oh, that's a great question. The simple answer is no, that doesn't present a problem. The only targets that we discourage are targets that actually break bonds in DNA. If the target includes a DNA binding domain, we can actually use that to our advantage. We have algorithms that seek out both known and unknown consensus sequences of DNA binding domains. So that's not an issue.
Okay, thank you, Barry. And then I'll come to the next question for Olivier. Is it possible to use PDX models for all kinds of, all test items? And what about all different kinds of targets?
Yes, for sure. For some specific targets, it should be difficult. So for targets that are expressed by the cancer cell, yes, we can use PDX for all the compounds. For compounds that are targeting the immune cell, we speak about that during the webinar. So it's possible to humanize mice. The main difficulty, I would say, is to use PDX models that are to test compounds that are targeting the fibroblasts, for example, because all the fibroblasts that are infiltrating and that are constituting the stroma of the tumors are from mice and not from humans. So if we have a cross activity of the target between species, so it's possible, but if not, it's not possible to use PDX. But we can use PDX with other techniques, I would say, that was not possible to present here. But we can use ex vivo assay, in which we can use tumors directly coming from patients. And we can maintain the fibroblasts and the stroma coming from the patients.
So now do we have time for any more questions, or should I pass it back to you?
Would you like to do one more question? I think we can put one more question in.
Yeah, okay. I think there's another one for Barry. I suppose with your experience now, what would you say would be the biggest challenge when applying the DEL technology to hit finding, and what kind of message would you give to companies potentially looking into this approach? What are some of the things to keep in mind and to be aware?
Well, the biggest challenge for this technology, pretty much to any hit-finding technology, is our understanding of the target. The better that you understand the target and the folded ensemble that it presents to an encoded library, the more likely we are to succeed. Highly mobile proteins are a challenge, just like they are for conventional high-throughput screening. Again, as I showed in the example, if you can focus the folded ensemble to a form that is relevant to your desired output, then you're more likely to succeed. So when we used that cofactor, you saw that that focused the folded ensemble. So understanding the protein is the biggest challenge.
Thank you.
Okay, well, thank you very much for answering those questions, Barry and Olivier and Emily, for conducting that. Now we're going to say goodbye. We have come to the end of the question and answer portion of the webinar. I know we weren't able to get through all the questions that we have, but I know the team at Oncology Design and HitGen will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that's on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box in a few minutes from now. You'll be able to view the recording of this event at that link and also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers again, Barry, Olivier, and Emily, for that very insightful and detailed presentation. So thank you very much.
Thank you, Sonia. Thank you, everybody.
Thank you, Sonia.
Thank you.
Thanks all for listening.
We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. Until next time, please take care. I'm Sonia Hunt, and bye for now. Bye, everyone. Thank you.
Bye-bye.
Bye.
Thank you.
Bye.